Great. Thanks everyone for joining this event organized by Single Cell and Spatial Consortium of Gulf Coast Consortium. We're a group of researchers from the neighboring nine institutions focused on single cell and spatial technology and its application to different area of basic and translational research. And for today, we're particularly excited to have a subject which focused on cancer vaccine study using different type of technology. And before I pass over to Dr. Prissy Ganarani, just say please follow us from the website. This is a GCC Single Cell Spatial Omics Consortium. We have a lot of activity, including both weekly seminar as well as the annual symposium, which is upcoming in October 22nd. Please feel free to register those events. And we also welcome new faculty and also junior research members to join the consortium. You can just find more information from our website. Okay, I'm just gonna pass over to Dr. Ganarani to host those events. And over to you, Prissy.
Thank you, Ken. And thank you everyone for joining us today. I'm Preeti Gunaratna, a professor in the Department of Biology, Biochemistry, and at the University of Houston, and director of the UH Sequencing Core. I'll be serving as the moderator for today's workshop. And as Ken indicated, as you all know, utilizing the body's immune system to specifically target and destroy malignant cells using immunotherapy has substantially improved clinical outcomes for cancer patients. However, significant challenges, including immune resistance, tumor heterogeneity, immune suppressive tumor microenvironments, have failed to bring these benefits to poor responders. So today we invited 3 speakers who will share with you their work on advancing this front using cutting-edge single-cell sequencing and spatial omics technologies. Xianli Zhang will share her work on multivariable regression modeling to determine baseline features required for major response to induction dual immune checkpoint blockade. And Sakuni Rankot-Gedura and Anjana Bhardwaj will share their work on developing peptide and mRNA-based vaccines to generate robust, long-lasting immune responses against tumor-specific antigens, a field that is emerging as a frontier in personalized cancer treatment and prevention. So I would like to invite the first speaker, Dr. Sakuni Rankot-Gedura, is a postdoctoral fellow in the Department of Genetics at MD Anderson Cancer Center, where she combines single-cell RNA sequencing, xenium, spatial transcriptomics, flow cytometry, immunoassays to uncover chromosomal missegregation that drives MHC1/2 antigen presentation and immune clearance in mouse intestinal epithelium. She earned her PhD from the University of Houston while serving as the lead scientist at the UH Sequencing Core, where she oversaw the production of over 5,000 NGS libraries, including single cells. So Sakuni, take it away from here.
Thank you so much, Dr. Gunaratna, for that introduction. I hope everyone can see my slides. Hello, everyone. Thank you so much for joining the workshop today. So my session will focus on how to dissect the neoantigen-specific responses of CD8 T cells to the fusion CD74 NRG1, which we detected in lung cancer. So to move forward, now looking at this personalized cancer vaccine approach, our focus is when a patient walks into the clinic, we sequence them, identify the patient's specific mutations, and delineate the best epitope targets for a cancer vaccine out of those mutations through a bioinformatic pipeline. And then we design and process the peptides for these epitopes that we have selected and deliver them to the patient through a suitable mechanism. And last but not least, monitor the immune responses after administration of the vaccine to that patient. So when looking at a cancer vaccine, there are like several major outcomes of a cancer vaccine. And one of like the most major outcome is to activate the T cell immunity within the patient to localize the immune response on cancer cells and to target the cancer cells via the patient's own immune system. And obviously, to do this, it is highly important that we select the targets properly so that they have a rich antigenic potential to be recognized by the T cell through these various inbuilt co-stimulatory processes. Now, in the context of a cancer vaccine, we have three main antigens that we need to discuss about. So there are self-antigens. These are basically part of our own body, and we avoid them to overcome any effect of autoimmunity. And then we have tumor-associated antigens. These antigens are self-antigens. However, they are differently expressed on the tumor cells. They're highly prevalent in the population, and they're widespread in the use of different immune therapeutics. And coming to the tumor-specific antigens, these are also called neoantigens or novel antigens. And this is the main focus for today's talk because these are highly specific to the tumor cells. Now, our source of neoantigens, it comes from fusion proteins. So fusion proteins, they can be translated from a chimeric RNA that was resulted either by a chromosomal rearrangement at a genomic level or a splicing error that took place at a transcriptomic level. And when detecting these chimeric RNAs, there are several different methods of chimeric RNA detection. And one method of high confidence is RNA sequencing. So when detecting chimeric RNA through RNA sequencing, we utilize the discordant paired end reads. So any fusion that is supported by discordant paired end reads, and especially reads that are junction crossing reads, which basically overlaps the junction of a fusion where gene A and gene B fuses together, these we consider to be fusions of high confidence and also of high interest due to their high confidence. There are like several discovery tools in the field today where you can use to detect chimeric RNA. Like Ariba, which is a well-known tool for chimeric RNA detection. Now, if we look at the central hypothesis which led to our project that is in discussion right now, so basically within a tumor and the microenvironment of the tumor, there is a high population of neoantigens. And we saw that the fusion junctions of chimeric RNAs, these are a good source of neoantigens where you can design peptides right from the neoantigenic region where one gene fuses with another gene. And these targets can be used for both the generation of peptide cancer vaccines as well as mRNA cancer vaccines. And then for today's talk, I'm going to specifically focus on CD74-NRG1. This is a known cancer driver and is also actionable because right now in the field, this is a highly attractive target. And there are several monoclonal antibody therapies used to target the fusion to control the downstream cell signaling pathways that are leading to cell proliferation, the uncontrolled cell proliferation. However, it's also a widespread fact that these monoclonal antibodies are not of high efficacy as we want them to be. So that is why we were focusing on how to enhance the immune therapy for CD74-NRG1 using cancer vaccines and also alternative immunotherapeutics. Now, for all the validation assays that I'm going to show you today, we have used two PDX models, which were given to us by one of our collaborators, Xenostat, and both these PDX models are made of a tumor coming from two patients having non-small cell lung cancer. So this is the ARIBA output for the two PDX models. And as you can see, for both PDX models, the fusion sequence, the fusion transcript sequence was consistent. And CD74 came from chromosome 5, NRG1 came from chromosome 8, and there were 6 exons joining from CD74 to the 5th to the 12th exon of NRG1. And with this transcript in hand, we were able to put together the full fusion sequence for the fusion. What's highlighted here is the fusion junction, which we are going to give more focus on. And then we designed 2 primers flanking this fusion junction, which were used for a PCR validation, followed by Sanger sequencing of these PCR products. So once we have validated that this fusion is actually present in the PDX tissues, the tumor tissues, we went ahead with putting together the fusion protein sequence. And as you can see here, this is the OFF, that was the longest OFF. And in here, the amino acids shown in red came from CD74. The ones in blue came from NRG1. And what's highlighted here is the fusion junction of the protein sequence we used to design our peptide targets from. And the one amino acid that is shown in black here, this was actually a unique amino acid due to the way the OFF was put together. Now, taking this fusion junction sequence, I designed the 9, 10, and the 11-mer peptides, which went into all the downstream analysis pathway that I'm going to talk about today. So there were 27 neopeptides of interest for this project. And we also looked at what HLAs the patients who were used to design the PDX models comprised of. So for this, we used OptiType, a bioinformatic tool which can be used to identify the HLA genotype of a patient using RNA sequencing data. And what's shown here are the HLA classes we identified in the 2 PDX models along with their coverage maps. Now, with the 27 neopeptides in hand, we also used 12 wild-type peptides which were designed either from the CD74 region or the NRG1 region. And these were tested against all the HLA classes you just saw before using these in silico tools. All these tools, they predict the affinity between the peptide and the MHC molecule. Now, we saw that HLA B702 showed a higher affinity out of all the HLAs that were selected, and these are the peptides that were actually predicted by the 3 tools I showed you before. And as a summary, these were the number of peptides we started off with, and then we ended off as positively binding to HLA B702. Then we saw that we needed to validate this prediction with the in vitro flow cytometry assay. And for this flow cytometry assay, we used eCMERS, where the 39 peptides you saw, along with several other control peptides, were loaded onto these eCMER peptides. And with 2 appropriate controls, we looked at the intensity of binding of beta-2 microglobulin to these complexes. More stable the complex is, the signal was highly amplified, and less stable the complex, the intensity of the signal was low. And we had several controls for voltage gating, and that way we saw that out of the eCMER analysis, there were 9 peptides that were validated to bind to HLA B702 through flow cytometry. And when compared with our prediction tools, these were the success rates of the prediction tools. So now that we had validated 9 peptides to actually bind with HLA B702, we are going ahead with the immunogenicity assessment of these peptides that are validated to bind to the HLA class that we want. So for this, we did an ELISpot assay. So for the ELISpot assay, we used HLA-matched PBMCs. These were actually incubated with the peptides of interest. They went through 14 days of stimulation with the peptides. We isolated the CD8 T cells from the PBMCs, and then they were put through a IF and gamma ELISpot assay. And then with the ELISpot assay, we saw that, so we had 45 peptides in total, which were our control peptides, our neopeptides, and wild-type peptides. There were 16 peptides that were showing a positive immune response. And out of these 16 peptides, the 7 peptides were actually neopeptides. And these 7 neopeptides are shown in the green arrows. So when looking at the ELISpot result, so these peptides that we saw in the ELISpot, so these 8 peptides actually uses that wild-type peptide as well. You can see that there was a match of 7 peptides coming from our binding affinity tools. And with these 7 peptides that were actually validated from the immunogenicity assessment, we went ahead with the investigation of the antigen-specific T cells and the T cell receptor changes that were caused by the antigen presentation using a dextramer assay. So a dextramer assay is actually a ECMR assay or a tetramer assay put together with single cell sequencing. So how we performed this was we had our HLA matched ECMers. These were loaded with our peptides, the neopeptides, our control peptides, and then they were actually barcoded with a 10x genomics compatible barcode. So this entire complex, it also had a fluorophore on it. So this fluorophore, we are able to detect it through flow cytometry. So what we did was we incubated PBMCs with this complex, and we did a flow cytometry analysis where we were able to sort the cells that are positive for the dextramer. And these cells were put together a gene expression analysis and a T cell receptor clonal repertoire analysis using 10x genomics. Now, there were several challenges that we faced during the dextramer staining. So one was to actually decide on how to identify the false positives. So for this, we actually used a dextramer negative control, where what we did was we incubated our cells with the Ebola epitope as well. And we know for sure that these cells were not exposed to the Ebola epitope. So anything that came up positive in our 10x genomics analysis, those were considered to be the false positives. So these were the 9 populations of the T cells that we identified through the 10x genomics gene expression analysis. So these 9 populations, if looking at the different cell states in depth, so what we actually wanted to identify was, out of these cells that we have detected from our 10x genomics analysis, what percentage of them are actually activated. So we have naive T cells. These they do not show, like they were exposed to the peptide or they were not exposed to the peptide, but they didn't show any response. And any cell that was activated by the exposure to the peptide, they either go through exhaustion due to the high amount of cytokines that they are producing, or they become memory T cells, which in turn become effector memory T cells. So within all the populations that we were seeing, we were interested in looking at the naive T cells, the activated T cells, and the terminally exhausted T cells, the precursor exhausted T cells, and the effector memory T cells. And these were the proportions of those T cells that we identified in all the cells that we were able to see in our 10x genomics analysis. So if we look at these proportions in a peptide-wise analysis, we saw that there were 3 peptides that did not show any activation at all. And these peptides actually did not show a positive immunogenic reaction in our ELISpot. And this particular peptide, the ECMR flow cytometry analysis, did not show any positive binding affinity. But other than that, there were 13 peptides that showed a potential of eliciting an immune activation to the CD8 T cells based on the presence of early activation CD8 T cells, effector memory CD8 T cells, terminally exhausted CD8 T cells due to the overexposure, and the precursor exhausted CD8 T cells, which were just being exhausted by the response. So with that, we concluded that out of the 39 peptides which were associated with our entire fusion junction, 6 peptides were actually giving rise to antigen-specific T cell activation. And there was one other layer of information that we were able to get out of the dextramer assay, and this was looking at the amino acid sequences of the T cell receptors that were being activated by the peptides that we used. So these were the amino acid sequences, both alpha chains and the beta chains, that were responding to the peptides that we used in the dextramer assay. So why is it really important? So this actually has the clinical significance that if we want to couple our cancer vaccine along with TCR T cell therapy to give another boost to the cancer vaccine, we can actually utilize patients' T cells, clone them with the T cell receptors that are responsive against the neoantigenic peptides, and increase the CD8 T cell response towards the cancer cells by putting these cells back in the body of the patient. So this is basically the process of how to clone the TCR to the T cell and then reinfusing it to the patient. And with this, I end my session, and I thank everyone who contributed to the project, who helped me along the way, and especially Gulf Coast Consortium for organizing the workshop today. Thank you very much. I would like to take any questions if you have any, and then if we don't have enough time for all the questions, you can also send me an email regarding them. Thank you very much.
Thank you, Sakuni. I think it is the case we are tight on time, so I would really encourage people to put their questions on chat, and we'll keep, address them at the end if we have any time, but ideally send the questions on email to the speakers. Thank you, Sakuni. Dr. Xianli Jiang is a postdoc at MD Anderson Cancer Center, specializing in computational immuno-oncology. She earned her PhD on computational biology from the University of Texas at Dallas. And she has a strong research focus on understanding T cell receptors and their antigen specificity, identifying therapeutic T cells and vaccine targets. And today she will present her work.
Hello. Hi, can you see my screen?
Yes.
Okay. Hi. Good afternoon, everyone. My name is Xianli, and I'm a postdoc from Dr. Ken Chen's lab. And today, I'd like to share our current findings about Treg effector Treg depletion reinvigorates the HPV antigen reactive T cells and drive the response to dual ICB in HPV positive cancer. This study, it's a large scale single cell profiling of a clinical trial of combination ICB by targeting the PD1 and the CTLA4. And we have collected data from baseline and the end of cycle one of this dual ICB. And we have the single cell RNA-seq data and the single cell TCR data. Our goal of this project is trying to understand the tumor immune microenvironment features that could mediate the response to the ICB treatment, and also that related with the clinical outcome of this response of this treatment. And the second goal of this study is trying to find the therapeutic T cells and antigen specific and their antigen specificities, which is still ongoing. And first, I would like to share the brief pattern of our patients. Our patients have around 46% of the responders, which show tumor reduction in the tumor viability in histology. And also the cell have reduction in the cell-free HPV amount. And the other patients are considered as non-major histological responder. And we first find our responders will have a reduction in the malignant cells after the treatment. The proportion of the malignant cell can be as low as 0% at a single cell level, and it is correlated with our the finding of the tumor viability in the histological slides. And we also find the after treatment, the responders will show increased CD8 or CD4 T cells as well as the CD8 to Treg ratio. So since the treatment is targeting the PD-1 and also CTLA-4, so in this study we're focusing on the T cell profiling. And we first focus on the CD4 T cells, and we've identified very highly activated Treg subcluster, which we named as effector Treg, and it shows the upregulation of CCR8, 41BB, CD25, CTLA4, and OX40 gene expression. All those genes are related to the immunosuppressive function of the activated Treg. And we find there is a dramatic reduction in this ETreg subset in after treatment, and this reduction is only observed in the responders. And on the other hand, this ETreg reduction, or we call it depletion, will happen only in the ETreg cells with high CTLA4 expression. So the figure on the right panel showing that the depletion will, there will be more depletion for the Treg cells have higher CTLA4 expression. And it suggests that this depletion is probably mediated by the anti-CTLA4 ICB. And we also look at the CD8 T cell compartment. We are able to find several tissue resident memory CD8 T cells, which will show high, those TRM 1, 2, 3, 4, they will show high cytotoxicity score, and also will have high tumor reactive score. And also those 4 TRMs will very exhausted. And our own current project, which focusing on the tumor, the HPV antigen reactive TCRs study, our current study focusing on the HPV antigen reactive TCRs, and we find those HPV reactive T cells, they were enriched in the TRM phenotype, which are in the green color, but our control, which is called CEFX, those T cells containing the TCRs that are specific to the non-tumor related virus antigen, such as flu or EBV, they are enriched in the non-TRM, non-tissue resident memory state. So this data suggests that the tissue resident memory CD8 T cells, they are widely found in the in our TME, and also they will be cytotoxic, and they should be tumor reactive. And also it's ongoing study that we are able to utilize this the comparison of HPV T cell positive HPV reactive T cells versus the CFX T cells to generate a machine learning model which can help us predict more HPV antigen reactive T cells. And those predicted HPV antigen reactive T cells, they are mainly located in the TRM region, which this data further support the tumor reactive phenotype is also the other TRM states. And we also find that the change of several CD8 clusters, they are associated with the depletion of the Treg cells. First one is the effector 1, and then the TEM1, TEM2. They have the negative correlation, which means the more Treg are depleted, the increased proportion of those effector or effector memory phenotype CD8 T cells. But meanwhile, the TRM4 are negatively correlated with the ETRAG depletion, or are positively correlated with ETRAG depletion, and they will we observe that this most exhausted TRMs will decrease in the responders, but in a little bit increase in the non-responders. And we utilize the single cell TCR data, trying to check the change of those CD8 T cells. We first observe that there will be more CD8 T cells expanded or contracted in the responders than the non-responders, which suggesting the CD8 T cells in the non-responders will be more stable in terms of the clonal type size. And we also measure the clonal type of state transition in the responder and non-responders. We observe there's a higher transition index in the responders, suggesting the CD8 in responders, they are more likely to change their phenotype after treatment. And when we further look at the phenotype of the expanded TCR clonal types, we'll find that those expanded CD8 TCRs in MHR are more enriched in the effector 1 and the TEM after the treatment, which is consistent with our observation in the increased proportion of those subsets. But in the non-responders, this increase expansion is more happen in the TRM phenotype, which suggesting this the treatment-induced expansion is more stay within the TRM phenotype. So it's this current difference will suggest the ICB will actually acted in a different manner, and probably that is a consequence of the ETRAG depletion. And in contrast, we also observed look into the phenotype of the contracted clonal types, and in the there will be more contracted clonal types found in the image responder, and those contracted clones are mainly resided in the TRM cluster at baseline, and they will become effector or less exhausted TRMs in the responders. And we utilize the clonal type, the CDR3 beta and the CDR3 alpha sequence as the clonal type ID, trying to check the potential state or the phenotypic transition present after the treatment in the expanded clones. So we can observe that the phenotypic transition in the responder, which is MHR, they are more like TEM centric and also TRM centric. And after the treatment, they will become less exhausted. We'll observe the expansion of this the lighter green color state, which are less exhausted, and the cytokine producing, which is a red color. But in the non-responder, it is more TRM centric. So we observe this the transition from TRM to proliferating, which suggesting a very a classical role of the ICB, which will classical role of ICB will trying to activate the T cells or make the T cells more proliferated. And as a conclusion, we observe there's more. We observe there's more transition happen in the responders. And our RNA velocity analysis also confirms such transition. And we experimentally validated 3 expanded TCRs to be HPV antigen specific, and 2 from responder and another one from non-responder. And this the state their state distribution at baseline and on treatment kind of show the consistent with the observed transition pattern in the MHR and non-MHR. And also we able to check the clonal type, the potential phenotypic transition in the contracted clones in the MHR and non-MHR. In the MHR responders, the transition again is TRM centric, and it will from the least most exhausted phenotype to the less exhausted or to the cytokine producing state. But in the non-MHR, it's not TRM centric. And we also we're trying to from now until now we're able to find the potential consequence of the ETRAC depletion, and we also look at the other data, trying to find the reason of the ETRAC depletion, and we find the CD16 positive NK cells might be the reason of the ETRAC depletion, because we observe there's higher amount of those such cells in the responder at the baseline. And also we find that the patients who progressed, they will have at least a proportion of the CD16 NK cells. And this CD16 NK cell actually is among the top TME features that can be that in the multivariate regression model. And so as a conclusion, this study tells us that how the depletion of Treg happens in the responder. That is because the presence of the CD16-19 NK that which will bind to the anti-CTLA-4 drug, and will induce the killing of those ETRAG cells. And without the ETRAG, the CD8 T cells, which are the tumor-active CD8 T cells, will undergo the a reverse state transition, which will make them less exhausted and even like cytokine producing. But in the non-responders, which there's less CD16-19 NK, there will be no ADCC happen. So the function of the ICB will be classical, will be only targeting the TRM cells. And so this result will suggest that we can in the future we can develop ETRAG depletion related drugs such as anti-CCR8, which together with the immunotherapy or even with some the T cell therapy. And in addition to this implication, we are also trying to utilize the HPV antigen specificity data, trying to develop some public HPV antigen, which can be used as the therapeutic vaccine. And also we are able to find several TCRs that or TCR motif that can be found in several different patients, can be used as a T cell therapy. And so in the end, I would like to thank to our collaborators and and also my colleague in my lab, and also for organizing this seminar. So I'm happy to take any questions.
Thank you, Jianli. And we have only 2 minutes, so I would like to take this moment to thank all 3 speakers for 3 very exciting talks, and encourage everyone to reach out through emails for any questions. Thank you all for attending.